AHPBA. And thank you very much for joining. We have an exciting lineup of world-renowned speakers this evening to focus on endpoints within clinical trials, told within the context of trials that our speakers have participated on and worked on for ongoing trials. One goal of the clinical trial committee within the AHPBA is to disseminate and educate on clinical trials to hopefully increase the proportion of trials led by surgeons. And again, as I mentioned, this is the second of 4 installments on webinar series to hopefully do just that. Our committee is led by Dr. Sep Galami and Dr. Paul Kara-Nicholas, and we're looking forward to bringing the future webinars. So look out for in your email for the opportunity to sign up for those in the future. My name's Tim Newhook, and I'm a hepatobiliary surgeon at MD Anderson Cancer Center, and I'm excited to moderate this webinar with my partner here, Dr. Akil Chawla, who's a surgical oncologist and pancreas surgeon at Northwestern Medicine in Illinois. Without further ado, turn it over to Dr. Akil Chawla.
Thanks, Tim. It's my pleasure to moderate this session with you. I'm really excited to have this great lineup of speakers. We have 2 outstanding clinical trialists and investigators to discuss endpoints with us today. And I'll turn it back to you to introduce our first speaker.
Thank you. It's an absolute pleasure for me to be able to introduce Dr. Hap Tran Kao. Dr. Tran Kao is a hepatobiliary surgeon and surgical oncologist, as well as a specialist in robotic surgery here at MD Anderson Cancer Center in Houston, Texas, and my esteemed partner. He has tremendous experience, and particularly ongoing experience currently with clinical trials focusing on therapy sequencing for patients with intrahepatic cholangiocarcinoma in particular. Dr. Tran Kao went to UCLA for university, followed by just traveling a little south to UCSD for medical school, followed by residency at the same institution. Following that, he completed fellowship in complex general surgical oncology at MD Anderson Cancer Center, after which he went on to his first position at Baylor across the street. We spent the next 4 years or so scratching and clawing to bring him back at MD Anderson, which he joined the faculty in 2019. Really excited to hear him discuss in particular one of his trials focusing on cholangiocarcinoma. Without further ado, Dr. Tran Kao.
Thank you very much to all of you for the kind invitation and for the very kind introduction, Tim. So for the talk today, I kind of actually wanted to really dive a little deeper into the NCI-ETCTN process, which I think a lot of surgeons don't know about, but have since learned about a little bit. And I think it's worthwhile highlighting this process. And to do so, I want to just use one of the trials that, as Tim said, I'm participating in neoadjuvant therapy for high-risk resectable intrahepatic cholangiocarcinoma, being run through the NCI-ETCTN process. So I'll give you a background about the trial first. We know that outcomes for patients with intrahepatic cholangiocarcinoma as a whole are pretty poor. And then when you have high-risk features, which I'll go into, they're especially poor. And if you look at both a number of retrospective institutional series as well as national databases, the data seem to certainly suggest that a neoadjuvant approach, when possible and completed, by that I mean the patient can get through neoadjuvant therapy and can get surgery, may be associated with improved survival. We know based on TOPAZ-1 that GemCis-IO is now standard of care first-line therapy in the metastatic advanced unresectable setting. Keynote-966 showed this to be the case as well, with pembro subbing in for durva. So on the heels of TOPAZ-1 and really concurrently, we had suggested, proposed a hypothesis that perioperative GemCis-durva for patients with resectable iCCA but high-risk features not only is safe, but can also improve outcomes compared to historical standards. And I want to take a moment to really kind of go over the endpoints, because after all, that's what this webinar is about. And I will highlight to you why some of the endpoints may be different from what we set out at the beginning to show. So for instance, the primary endpoint in this trial was proportion of patients who would complete 4 cycles of neoadjuvant therapy and surgery. If the patient was to progress and not be able to get surgery because of disease progression, that would be considered a failure. And I'll go over why this is the case and why, for those of us who treat this cancer, it's not really the concept that we really follow. For all of us who treat high-risk cancers like pancreas cancer or cholangiocarcinoma, we know that in the presence of high-risk features, the use of neoadjuvant therapy, one of the indications for it, is for allowing the biology to declare itself and letting the disease course kind of prove itself, right? To better select, ultimately, patient selection for those who will stand to benefit from surgery. However, the ETCTN is run through the NCI by a group of scientists and clinicians, some of whom are not in this space necessarily, who basically are very concerned about the quote-unquote missed opportunity for surgery, right? So in other words, if you could operate on them now, why would you wait and put them through neoadjuvant therapy? And so after much debate and deliberation, we had to acquiesce to the idea that we couldn't hurt patients or reduce their chances of having a curative intent operation. So in this case, primary endpoint, as I said, is proportion of patients who successfully complete 4 cycles of neoadjuvant therapy and surgery. The secondary endpoints are things like recurrence-free survival, overall survival, which ultimately is what we all care about. There was an interest also in proportions of R0 margin resection, response rate, major path response, defined by less than 10% viability, as well as adverse events, and being the ETCTN, a number of correlative endpoints that I will also go over. So the study design was for patients with potentially resectable intravertebral cholangiocarcinoma with high-risk features, being tumors greater than 5 centimeters, multifocal tumors, major vascular invasion, regional nodal disease, or elevated tumor markers above 200 units per micrometers. It's a single-arm study studying neoadjuvant gemcitabine durva, followed by resection, and then adjuvant gemcitabine durva for another 4 cycles. And the comparator in this case was single-arm historical kind of control. And again, the outcome I just highlighted to you. So these are patients who have to have resectable disease, histologically confirmed intrahepatic cholangiocarcinoma, technically resectable on imaging evaluation and by a surgeon trained in AHPBA or surgical oncology. It has to have high-risk features, certainly good performance status, treatment naive, and no autoimmune condition, of course, because of the use of immunotherapy here. So the high-risk features, again, have been listed before. These were the same features except for the tumor marker that we used for the NEO-GAP trial that Shishir Michael chaired. So a lot of the same risk features that have been shown in a number of studies in the past to be high-risk predictors or factors. So here's the study schema. Again, patients with resectable disease will go through 4 cycles of neoadjuvant therapy with gemcitabine durva. The primary endpoint in red here is the proportion that gets through that and gets to surgery for curative intent resection. Secondary endpoint are in blue. So again, major path response, those who can get the adjuvant therapy, adverse events, survival, overall and recurrence-free, as well as proportion of R0 resection. There are a number of exploratory endpoints that I'll go over, and then the rest of it is just treatment, the treatment paradigm. So for statistical consideration, if you look at currently available data, the success rate of this approach, the null hypothesis is 65%, so we predict that at least 85% should be able to get through this. Given this, we had, this is going to be a 2-phase study. Total target sample size is 27. We're going to pause at 12 patients. We have to make sure that at least 8 of them get through the neoadjuvant period and to surgery. And if they do not, we will have to suspend the study and reevaluate. Now, here's the nice thing about it is that when we do reevaluate and sit together and go back over the patients who, let's say, failed, we then can go back and debate and deliberate about that high-risk population where now you're allowing for sparing them a futile operation. Some of them might have had indeterminate lung nodules that over time have grown and show themselves to be metastatic disease. Ultimately, though, we want to get up to 27 patients. The correlative studies are here. Essentially, we're going to collect these at multiple time points. This is a pretty heavy correlative type of study. We're going to look at RNA sequencing, looking for gene expression profiling, and into the immune cell composition. The hypothesis being that neoadjuvant chemoimmunotherapy can be associated with increased interferon-associated signaling pathways and cytotoxic immune cell infiltrates. We'll look at whole exome sequencing to evaluate the tumor mutational burden and loss of HLA heterozygosity, because our, of course, idea, the hypothesis is that the higher the tumor mutational burden and retention of your HLA prior to therapy will be associated with better response to this neoadjuvant regimen. Multiplex IHC and IF to look at spatial distribution of your tumor-infiltrating lymphocytes, and as well as immunosuppressive cell types. We'll have mass cytometry, looking at the circulating immune milieu, looking at functional T cells, as well as suppressive cells. And then ctDNA, exploring the role of ctDNA in assessing MRD, which has to this point really not been proven for biliary tract cancers, except for small kind of studies. So, in summary, for the trial, there's a strong rationale for us to use neoadjuvant chemoimmunotherapy in the treatment of iCCA with high-risk features. This is a multi-center trial, phase 2, to evaluate the safety and efficacy of this approach. Multiple correlative endpoints will reveal novel immunologic and microenvironmental biomarkers of response to therapy, and ultimately will also help to clarify the role of ctDNA in the evaluation of MRD. I want to talk real brief about this ETCTN process. I've had a chance to talk to a few surgeons about this once they heard about this, because they wanted to learn more about how to get their trials through this. This is actually a really neat thing. A lot of people know that CTEP, the Cancer Therapy Evaluation Program of the NCI, is composed of the NCTN, which we're all familiar with, that SWOG, Alliance, ECOG-ACRIN, as well as a Canadian network group, as well as a children's oncology group. But it also has a couple of other trials network, one being the pediatric early stage clinical trials, and the other being this one, the Experimental Therapeutic Clinical Trials Network. And the two people I show here are the people at MD Anderson who've introduced me to this and have allowed me to kind of pursue this pathway, which has proved to be very eye-opening and frankly, a great opportunity for surgeons, I think, who want to be involved in perioperative and neoadjuvant trials and novel approaches, potentially resectable disease. And it is really a partnership between the NCI, pharmaceutical industry, academic institutions, and individual investigators for early clinical evaluation of innovative cancer approaches and therapies. A couple of their objectives are to integrate preclinical findings, using clinical samples for biomarker analysis, and to integrate molecular characterization, cancer biology, and imaging into clinical trials. So lessons learned from me in terms of clinical trial endpoints, and I think most of you are well aware of this. Endpoints, of course, will depend on the type of trial you're running, whether it be phase 1, 2, or 3. But more so, what's funded will depend on the funding source. The ETCTN is very heavy on correlative studies because they want to understand the mechanistic underpinnings of response and non-response. Conversely, I have a industry-sponsored trial. They're much more interested in looking at, clinical kind of outcomes like path response, right? So that they can that's what they're banking on rather than the correlative stuff. They're not too interested in that necessarily. They have to be practical. So what do you care about in terms of your clinical trials, and what surrogate marker may be appropriate for it? We all care about overall survival, but to study overall survival as your primary endpoint will take a lot of money, will take a lot of time. And so you have to then decide what is the appropriate surrogate marker. Is path response an appropriate response, marker for this? I'm not sure, and that's something we need to evaluate and discuss amongst all of us. And finally, you should probably probe deeper than feasibility and safety, because showing that alone will not influence practice, right? How will your trial inform and influence practice, or how will it bring about new knowledge that can really drive science forward and allow us to build upon that knowledge to then be better at perhaps personalizing treatment for these types of patients? So it was a whirlwind, but I was told 20 minutes, so hopefully I stuck to that. Thank you so much. Happy to take any questions.
Thanks, Dr. Trancal. That was phenomenal talk, and certainly for a surgeon investigator, a unique method to open a trial through the NCI mechanism. Just some questions for you, just to start off the discussion. For the ETCTN, obviously very heavy on correlative studies, and given the design of your trial with the small number of patients we're really looking at, clearly the emphasis is on the correlative studies there. To what extent does the NCI really require a fully developed plan for each of these correlatives? Do you have to have a platform for each of these experiments set up, a correlative PI for each correlative and a statistical plan? Or is this similar to what we see for NCTN studies where perhaps the technology might evolve over a period of time, and it's in the best interest not to necessarily have that up front?
Yeah, that's a fantastic question. So now the NCTN process, of course, we're familiar with, and I'm going through that myself right now. The ETCTN, you're right, is incredibly focused, right? They do not have the bandwidth or the money to support 300, 400 patient trials. They just don't. They told me very specifically from the get-go, they're like, look, we're going to fund a smaller study where we can really start to study the mechanisms, the biology, the predictors, right? And so you're completely right. For you to have an ETCTN trial, you need to have a correlative translational PI as well. So I am the trial PI. I have a translational co-PI who is basically in charge of, thinking through and sharing these correlatives and demonstrating that they have the experience to do it. We're fortunate, of course, at Anderson that we have a lot of these people, and Tara Haymaker is my partner in this trial, and she's fantastic. But yeah, I think it is a focus mechanism for a reason, is because they are willing to spend the resources to get at these tissues.
We have a question in the chat here from Dr. Martel. Do you think your primary endpoint should be different depending on whether you are designing a single arm or a dual arm trial?
Yeah, I think that's a great question. Again, it depends on what type of trial you're running. Like, for instance, in this trial here, right, where we're looking at, let's say I'll compare it, if I had a second arm, it would be upfront surgery, right, rather than perioperative. Then clearly the primary endpoint will change, right? Because the primary endpoint is no longer going to be what proportion can get through neoadjuvant therapy and get to surgery. Primary endpoint in that case will have to be changed to something different, like, disease-free survival, progression-free survival, and then again, a lot of the correlatives because it's ETCTN. So I do think that whether you have one arm or two arms does dictate what kind of endpoint you ultimately have to study.
Just to follow up on that, I have a question myself, which is interesting mechanism that is available to surgeons to use and utilize. But you mentioned that clearly funding, which is always a huge issue, which we'll get to in another webinar. But the funding through this mechanism is clearly very tight for them able to offer this resource broadly. And how does that and did that impact your selection of an endpoint? Because clearly, if you chose an endpoint of proportion of patients who live 10 years, you're going to require a ton of cash, right? So when trying to think about how to answer your question, when you're mulling this over, did your choice of endpoint be one such that you could have a nimble and dynamic trial for those reasons?
Yeah, no, absolutely. You have to take that into account. And I think one thing that was really eye-opening about this experience is that there was a lot of back and forth. There was a lot of conversation, a lot of getting together and talking through, trying to share our expertise with this disease and explaining why it is that wouldn't necessarily be considered a fail, if you will. But then again, at the same time, acquiescing that that's going to be something that they're looking for. But the key thing is the allowance for there to be secondary endpoint, correlative endpoints, and the ability to get back to the table and discuss, right, down the line, how to adjust if needed at all, the study. So, I think it's been a really positive experience overall. I will tell you the ETCTN by and large is a mechanism that studies drugs a lot in the metastatic unresectable setting. I believe mine was probably only the second or something kind of surgical type of trial that they were able to fund or that they were willing to fund. But I think they didn't get that many applications anyway, and they were very open and they're very enthusiastic about supporting these types of perioperative trials. So something to look into for those of you who are working in that space.
Really interesting. Having gone through the ETCTN process, I think you're probably getting lots of questions from investigators all over the country on how that process went. You think about a committee like ours and the Clinical Trials Committee through the AHPBA, and you mentioned having a correlative PI. I envision a potential for investigators throughout the country that have their own expertise to come together to build a trial in a locally advanced or local regional space that potentially could enroll nationally, something similar to what you've designed. Is there space for that level of collaboration through this mechanism? Is this something that perhaps we should be thinking about pursuing through our organization?
Yeah, no, I would. So just so you know, before you can even consider ETCTN trial, you have to have the letter that states the support of at least 12 participating sites. Okay, so there is basically an expectation that this is going to be team science, that everybody's going to take part in this together. And so the first step was for me to develop the concept, to then reach out to a number of surgeons and centers that I know, and surgical oncologists and medical oncologists and centers that I know and that share, an interest in this disease like I do. And then to basically say, here's the trial I want to run. Here's the mechanism by which we're going to run it. They want support from at least 12 sites to say that they will open the trial. And you submit that list of names to them along with your concept. And that's just to get the conversation going with them. And that was, not a hard thing to do. And so right now we've got more than 12 sites opening this trial already. Vanderbilt just opened just yesterday. And so, it's been very positive experience overall. And I would encourage everybody to think about this, especially if you're looking to study the correlative aspects of it, the translational aspects of it. Understand this is not the trial that's going to allow you to study randomized double-arm, phase 3 standard of care versus, where you're going to need 200, 300 patients. This is a smaller kind of process, but for each patient that you recruit, you actually get more money than the NCTN, just so you know. So it's well supported at the institutional level once you do recruit the patient.
We have another great question from Dr. Martel. Did you include patient viewpoints when picking your primary endpoint? I think you mentioned clinicians, he says.
Yeah, great question, Guillaume. And so patient viewpoints, patient advocacy is now more and more, of course, taken into account when designing clinical trials, as they should be. This being a more, again, translational trial, that we didn't have the opportunity to do that. But definitely, as you start thinking through clinical trial concepts, you need to involve patient advocacy, especially if you're going to design a bigger type of trial.
I think Dr. Gholami has a comment.
Yeah, thank you so much, Hop, for this wonderful talk. I wanted to just make a comment for the listeners who are really just as their first introduction to trials, and not be mistaken to think that, that a safety and feasibility is not the way to start, right? Because you cannot just jump into an exploratory kind of thing. It really depends, right? Like what your setup is, what you have access to, right? Like who will fund it. And I think when you're studying something completely new, like I have an IIT that's a neoadjuvant trial, and we are starting with a safety and feasibility, because again, if it's never done in something, so it's not for cholangio, for example, my trial is something to do with colorectal liver metastases. But if you're never introducing another agent before that, then I do think that, you want to prove first that patients can get safely through that treatment, and then get to surgery, and then a safety and feasibility may be appropriate, you know? So just sort of like kind of putting into context that there are different types of sort, and that I know you're not trying to say that, but I just want to kind of like make that a teaching point, right?
I would say I applaud you for clarifying that. I would say the following, though: as you design a safety and feasibility trial, do look for ways to further supplement that. And by that, I don't mean like you have to have all these, you know, advanced correlative studies ready, but include things that allow you to better understand why it may not be safe in some patients, why it may not be feasible in some patients, and try to get at that. That's what I mean. But absolutely, these have to be proven before you do these big trials to ultimately try to challenge standard of care potentially.
Yeah, that's a good point, because I think the teaching here is also like, as you are essentially doing a trial and you're going through all that effort, right? Try to maximize. Now, don't make it too complicated, because if you're making it too complicated, then you're going to get a hard stop at feasibility, right? And then people are going to say that, well, like, you need this and X, Y, Z, and this is becoming too complex, where it cannot just be run at multiple centers, right? Like you're at Anderson, maybe some other centers don't have all the resources that Anderson has, right? So I think there's a fine line between, and a balance of how complex do you make your trial, but also try to get right the information that you're really interested. If biology is really what you're interested in, right, and you're starting with safety, like, yeah, try to get like the simple correlatives in. And I think that's a really wonderful segue to the next talk about translational sites. But thank you again for this great talk.
I'm just going to chime in for one more question. We talked a lot about getting the trial through the ECTN mechanism. From a standpoint of next steps for this study, obviously, we're going to see kind of what these correlative endpoints show. What type of hypotheses are you most excited about, and what are potential designs for a follow-up study? Since we have you here with us, we'd love to hear some of your insights there.
Yeah, no, that's a fair question. I mean, whenever you use immunotherapy as part of your treatment regimen, there's always going to be an interest in finding out whether we can predict, based on baseline, right, tissue biopsy or blood biopsy, whether we can predict who's going to really respond to that treatment. And ultimately, that is going to be possible, I think. I harken back to the trial that I, participated in with Dr. Ahmed Kassem in HCC, where we knew that, based on pre-treatment biopsy, if a patient has a low level of, tumor infiltrating lymphocytes, right, giving dual agents for IO was going to allow for an upregulation, more upregulation than single agent. And so that suddenly kind of gives you an idea that maybe in the future, if I biopsy the patient's tumor, right, and it's an immunosuppressive environment, especially immunosuppressive environment based on whatever parameters you want to measure, you can then decide this is not somebody who's going to do okay with just, let's say, Nivo, and they may need Nivo-Ipi, right, or things like that. So I think that's the thing. I think that ultimately this is driving at understanding what drives response and non-response, and therefore giving us lessons to learn from as we start to try to think about personalizing care in the future for patients based on their baseline tissue biopsy, based on their, what their blood, immune kind of milieu looks like.
Do you think that with 27 patients, you'll have enough subsets of patients to direct care?
I think we'll start getting a signal and getting some information. I in no way imagine that these 27 patients are going to tell us black and white, this is what needs to be done. But I do think it's going to start giving us some better insight into the relationship between the tumor, the natural environment and the natural immune system, and then the treatment.
Well, that's great. Really appreciate your insights. And I think we could talk all evening about this, but we do have to move on to our next speaker. I'm honored to introduce Dr. Zhenjian Ye. Dr. Ye is a distinguished surgeon scientist at the University of North Carolina at Chapel Hill. She serves as vice chair for research in the Department of Surgery at UNC. She's co-leader of the UNC Lineberg Clinical Research Program and director of UNC Lineberg Pancreatic Cancer Center of Excellence. Dr. Ye has really been a leader and is at the forefront of pancreatic cancer research. She's had a focus in applying genomics to identify novel target approaches for pancreatic cancer. Her groundbreaking work, I think we all know, has led to the identification of molecular subtypes for pancreatic cancer, which really has the potential to guide personalized therapies in this space. We're honored to have Dr. Ye join us today to talk about clinical trial correlatives. Thank you so much for joining us today, and please join me in welcoming Dr. Ye.
Thank you, Dr. Chawla. Thank you for that introduction, and I'm very excited by what the AHPBA Clinical Trials Committee is doing. I think that what you're doing is incredibly important, and I think that we don't necessarily recognize this as surgeons, but we really have key opportunities to really advance clinical trials and really, I think, sometimes think out of the box that may not be in that, randomized dual standard of care 2-arm trials, but we can really, I think I would put a challenge out there to people who are listening and will listen to the webinar, that we have an opportunity as a field to really accelerate bringing things to the clinic. And we should take every opportunity to do that, because I think our clinical trial systems right now are slow, right? And how can we be more efficient at some of what you've all discussed in terms of predictors, right? Because we all want predictors, and we're at a point, I think, in technology that we do have ways of leveraging some predictors, right? And so I'll talk about our molecular subtypes and sort of how that goes into how we think about that in terms of clinical trials. And I think that Dr. Tranco talked about this, right? I think that on his slides, if you notice, he talked a lot about the word integrated, and that's what a lot of his markers are integrated markers, right? Which are essentially retrospective correlatives, if you allow me to just simplify things, right? Where you do the clinical trial, you collect tissue, you do assays, and then once the clinical trial's finished, you then see if any of your assays or your tests or your hypothesis, and maybe you might discover new hypothesis from that, which will be great. If any of those will associate with an outcome that you're interested in, so that you can take the next step for another trial, right? So I will pose the question to the group and to the field: can we think about doing integral markers more often rather than integrated markers? And there are ways to do integral markers with enough sample size that one doesn't necessarily have to be essentially wedded to your integral marker, but you could actually be asking the question of whether or not your integral marker makes sense. For instance, you could be randomizing by the integral marker, right? Which is really, I think, the gold standard for testing whether a marker is predictive. Now, with many of the diseases that we take care of, it's hard, right? We don't have enough sample size. And so randomizing by integral markers sometimes, and when those integral marker populations are low, it becomes very challenging. But I think as a field, we should still think about it. And so I am going to talk about integral markers. All right. And so what are marker considerations? I think when you start talking about integral markers, we definitely start thinking about rigor, reproducibility, not that we don't otherwise, but I think it's incredibly important that whatever marker you're interested in, you show to yourself and the field that it's rigorous, it's reproducible, it's replicable, right? And of course, if it has clinical utility. Now, this is from a statistics write-up from a statistics blog, but I'm going to talk about reproducibility and replicability, because I think as a field, well, actually, until I really read this and would talk to statisticians, I never really understood the difference. I always thought they were interchangeable. But replicability, I think, is a higher bar than reproducibility, where you're using new data to get the same result, right? And that's what we really want when we're going into clinical trials, because you're going to have a new cohort. So in your new cohort or your next cohort, is your assay, or is your integral biomarker replicable? All right. And so before we go there, I would say probably many of you on the webinar are not old enough to remember this, but I'm at UNC and I'm a surgical oncologist. And when I first started, which was in 2005, there was a huge article that came out in Nature Medicine at Duke, down the street from us, where they reported genomic signatures to predict therapies. And it was huge. 3 clinical trials, multiple cancer types got started because of this, right? And so the hypothesis was that these chemosensitivity signatures that the groups derived could predict treatment response. And so 3 clinical trials started. All 3 terminated within 3 years because mistakes had been made with the data, and it was clear that this is actually not probably it was not chemosensitivity that was the signature. The data was retracted. And so I think that this really set the field back a lot, right? Because now we've been very gun shy about introducing predictors of response since this time period. And it's also made us a lot more wary, or a lot, focus a lot on rigor and reproducibility, and replicability, which I think is a good thing for us as a field. And there have been a lot of, I think, look backs as to how could this have been done better? What could we have changed? How can we make sure that future assays are more rigorous, right? And so this is if any of you read the Cancer Letter, it's sort of like the New York Times for cancer. And so these authors were saying that, the better predictors are the ones that are easiest to do. If they're not overly complicated, they're probably going to be the most successful ones. All right, so I'm going to give you our case use studies of how we came about an integral biomarker and the steps that we made in order to get them into the clinic. And so the beginning, right? So in 2015, and of course, this was work that was predated 2015 by a lot, but we had found subtypes for pancreatic cancer, and others have found it as well, right? And so one of the things that we do for replicability is always compare new findings and old findings and others' findings in the field against new data sets as well. So it's constantly something that we're doing. But for those of you who may not be familiar with this, what we did was we used gene expression data, and we found 2 groups. And when we looked at these 2 groups, the way that we did the analysis, we didn't know what the samples were. Okay, and these included primary, normal, metastatic samples, all from patients with pancreatic cancer. We didn't know what the samples were, and we definitely didn't know what outcome was. And so many times it's better to be lucky than good. And we developed a method essentially to hone in on tumor intrinsic signals. And I think that's why the biology, which I'll talk about a little later on, has been very successful and has been validated by many others. But the first thing that we found when we sort of unmasked all of our data was that one subtype, the orange basal-like subtype, had a much worse prognosis than the blue subtype, which we call classical, because Eric Collison, a medical oncologist that is now at Hutch, first published that there were 3 subtypes in pancreatic cancer, and one of his was classical, and so we called our classical as well. There's overlap. They're not redundant, but there was enough overlap that we wanted to pay homage to his. All right, and so the first question that we asked ourselves, and have been asking ourselves for the last many years as more data has come out and sequencing has become more and more prevalent, is are our findings reproducible and are they replicable, right? And so this is now on a new data set of 400 patients. We're still always looking back, still seeing the same prognosis. And I know others have seen this as well, which is incredibly reassuring. We're always sort of spying on others' publications to make sure that we didn't do anything wrong. And so, a nod to the current administration. So what about this fork in the road, right? So now we have subtypes, but what do we do about it, right? So how is it going to be useful? So in order to make subtypes useful as biomarkers, we talked about it being robust and replicable, right? But it also needs to be clinically relevant. And I think reviewers, probably even today, but definitely in 2015, said to us, is it really clinically relevant if you're able to tell somebody with pancreatic cancer that they have a shorter survival than another group, right? Without any sort of interventional options. Is that really something that's helpful? And so that has been top of mind and continues to be top of mind. And so one thing that we did was with help from many other groups, including Steve Gallinger and Jennifer Knox and Cao Yang, who's now in Texas, from Princess Margaret Cancer Hospital in Toronto. So we looked at their data on COMPASS, which was a fantastic trial that's still ongoing, where they were able to do molecular profiling on everybody coming into their trial, which was such a huge undertaking. And what they did was they just gave physician choice. And you can see that most physician choice was FOLFIRINOX, right? So this is their early data of their first 50 patients. And so we asked the question on the left, does subtype matter for chemotherapy response, right? And you can see that, these are the patterns where patients with basal subtype tumors grow. When you give them FOLFIRINOX, classical patients generally respond, that could be better to FOLFIRINOX. And then David Linehan, who, as you all know, is now dean at Rochester, was kind enough to share his samples from his FOLFIRINOX CCR2 inhibitor trial. And we looked at his data and found the same thing, where FOLFIRINOX plus CCR2 inhibitors, basal patients did not respond to them, and classical patients did. And so that begs the question, now do we have a clinical utility? Can we now predict response, right? Which is what the discussion was in the Q&A for just a few minutes ago. Right. So can we then, to predict response, can we then use an integral marker? And as we were thinking about this, right, this is when I learned what an integrated marker was and what an integral marker is, and realized that what we really wanted to do was to try and help with patient treatment decision making. And if we really wanted to do that, we need to somehow figure out how to put subtypes into the clinic as an integral marker. Okay, so what do you need for an integral marker? You need it to be in a CLIA laboratory. All hospitals have it, but not all hospitals have the same platforms and assays. So that's one thing. And if you're going to include this in a trial, you either need an IDE or an IND. And so we knew that the road was going to be long, and we started thinking about it. And so for integrated markers, I'm going to just step back 2 seconds. So a lot of groups have been looking at this over the past, I would say probably 10 years now, thinking about whether subtypes are important, using it as an integrated marker. So these trials are ongoing, right? And then you do subtyping with your favorite platform, and you go back and you look if there's any correlation response. And the reason why we're very confident that making this an integral marker is something that's important is because all trials are pointing in the exact same direction, where patients with basal-like subtype tumors do not respond to FOLFIRINOX unless maybe they have a DNA repair germline mutation or maybe a somatic mutation. In the PASO-1 trial that Jennifer Knox just presented in ASCO last year, where it's a randomized metastatic setting, FOLFIRINOX versus gem, nab-paclitaxel, same thing, right? Totally different trial, multiple institutions finding the same thing. So can we now accelerate this and move an integral marker into the clinic, right? And so the SWOG 1505 trial for neoadjuvant chemotherapy, dual first, 2-arm trial, GemNab versus FOLFIRINOX, no benefit, or I guess I would say equivalence, right? So we have equipoise, but our question would be to you, is there really equipoise if we start thinking about integral markers like patients with basal subtype tumors, right? And so how do you get an integral marker to the clinic? Well, first of all, you need to figure out an assay that works on a single sample. And of course, you need clinical utility. So hopefully I've convinced you that there's some clinical utility. And so now can we pick an assay? And then once you put it in a trial, right, this road is long before you get regulatory approval for using it as treatment decision-making. And I will say that this slide is not new. It's probably at least many years old, right? And we're on this, we're still on the first part. So long road. All right. So many of you who look at correlatives, right, will do proteomic analysis, genomic analysis, epigenomic analysis, panels of biomarkers, look at a heat map, look at associations. In general, what we're doing is we're comparing between groups, right? And sometimes we know what groups we're comparing. Sometimes we're searching for the groups that are important. But at the end of the day, comparing groups is actually not usable. So many of the heat maps that you see for clustering, great if you have a gene list and you're going to learn biology, but not really helpful if you're trying to bring it to the clinic, because it needs to be a single sample, right? So you cannot depend on what your neighbor is. You need to have a biopsy or a tumor sample sent to the lab, and there needs to be a call. Like there needs to be something that's reported out without having to go back to a heat map or a comparison. So that's the difficulty with a lot of the correlatives that we do. And so we reached out to a statistician who I still work with, probably about 10 years ago now, and Naeem Rashid, who's an associate professor at UNC, said, oh, well, for a single sample, what you need is you need a classifier, right? And so we were very fortunate that the classifier, remember what I said with the cancer, remember when I showed you the cancer letter recommendation, the single sample classifier is incredibly easy. 18 genes. All you have to do is look at whether a gene is high and low. You give it a 0 or 1, depending on whether the pair is high or low. You give it some corrections. You can even do this on a calculator. And then, figure out what the probability is. All right, so simple, right? Now, what assay can we use? At UNC, we've been really fortunate, and I think at MD Anderson, I know that you all have a nanostring assay too, and some institutions do, but not all institutions. But nanostring is an assay that can be placed into the CLIA space and validated. And so we reached out to our colleagues in Molecular Path and said, hey, can you please help us with this? We really would like to move this to the clinic. And we know that you already run the nanostring assay for breast cancer. So can we try and do this for pancreas? So we were very fortunate that they already had this running. And so it was more just validating a new assay on the same platform. So this is our CLIA validation. And what we did is we showed concordance. So the Cohen's kappa shows concordance between what we're using as ground truth, which is RNA sequencing, and then the new platform nanostring, which is really, it's very unbiased because it's just a direct count of the number of genes that are expressed in any single, for the number of expression, the number of counts of your genes in a given sample. Okay, and we did have 2 discordant samples that were repeated, and they were still discordant. And we think it's actually because they're in the middle. So they're sort of like, probably have a mixture of populations. And for those of you who are pancreas aficionados, it's basically saying that we have classical and basal mixtures. All right, so now we have an assay that's CLIA validated, right? So now can we use this in a clinical trial? And so Susan Tsai should actually be giving this talk and not me, because when she was at MCW with Doug Evans, she came up to me at an AHPBA meeting and was really interested in what we were looking at. And, you know, I think it really takes surgeons, right, to really move the needle sometimes. And so she said, well, can we just use subtypes to predict response? We have equipoise. We're not doing any harm. Then physician choice. Could we do better? So we said, okay, we're going to get this CLIA validated assay into the clinic. And the main, you know, the main issue was the turnaround time, but that's been great. That's been less than 5 business days. They've been able, they do it on US guided biopsies, and a report goes out from UNC Health to MCW and Honor Health, where Erkat Borzanski is running the trial for subtype, and basal patients go to Gemnat paclitaxel. Okay, so some warnings, I think, as you think about markers and assays, and for those of you who are developing correlatives. So your assay platform is incredibly important. I think many of us are trying to realize, are recognizing this as technology advances. There are so many different ways to try and look at the same thing. And the question is, are you actually seeing the same thing? And I would argue that with all of these commercially available platforms, just because your result is the same name doesn't actually mean it's the same result. And so that comes to replicability. Okay. We were looking at the Tempus platform. So UNC uses Tempus. And so we can just order Tempus, and now you can order Tempus Purus, but don't. So Tempus is a very interesting platform because it's really incredibly sensitive, and it's an exome capture platform, whereas RNA sequencing is a poly tailing platform, right? So you're picking up pieces of RNA and then sequencing it. And nanostring is you're just counting how many times your gene is expressed in a sample. And so when you do exome capture, it's like creating a probe, right? You want a probe to be incredibly sensitive. And so we compared head to head, same block, different sections between RNA sequencing Tempus, and we are seeing discordance, certainly still see some prognostic significance, but not as much as, not the drastic prognostic difference that we see with RNA sequencing and nanostring as well. Other groups are doing, looking at IHC and ISH. This is IHC, which may be different than gene expression, right? Because we're looking at protein. Susan's group, when she was at MCW, were nice enough to look at IHC, and you can see the concordance between a classical gene that's expressed versus a basal gene that's expressed with the current subtyping methods that we use. They're associative, right? But not exact. They're only right. They only agree about 60 to 70% of the time. All right. So those are just some caveats, I think, as we think about markers. And I think I'm almost out of time. So I'm just going to show you this other case use. So we've been asking the question so basal patients don't really respond to FOLFIRINOX. What else could they respond to? And we went through this whole unbiased proteomic discovery analysis and found that the kinomes in these two subtypes is actually very different. And while there are a lot of kinases that are really interesting, what really grabbed us was EGFR, because we thought that, shoot, there's been so many EGFR trials and they've all just been disappointing. Could we have missed something? Because basal subtype tumors are only 20% of the population, right? And so and now we're talking about sample size and power, is any clinical trial that's been done on EGFR actually powered sufficiently to see the signal in this 20% subgroup? So Eric Hollison actually introduced us to Stefan Bach and in Bach's group in Munich, who actually had just finished running 2 metastatic trials with EGFR inhibitors. One was Accept and Rash. Both were negative. So they were nice enough to give us their formalin fixed samples sections that we ran sequencing on. We were able to probably salvage about 70% of them. And so this is the data from the 70% of samples from the Accept trial. This was a 2-to-1 randomization, GEM and gemafatinib, with gemafatinib being the larger group, as you can see, right? So totally made sense. We didn't really select for a population, still seeing that 2-to-1 ratio, still seeing negative study. What happens if we sequence it and we ask what subtypes patients are? So what we saw, shockingly, was that basal subtype patients actually responded to EGFR inhibitor therapies, right? So with afatinib, we're actually, for the very, very first time, seeing equivalence between basal-like subtype patients and classical subtype patients, and the same thing with RASH. These are the HR ratios comparing both modern chemo as well as the accepted RASH studies, which are the 2 EGFR trials that we have. All right, so another opportunity, right, for an integral trial, and that's now to move to the next step. So if we think that basal patients may benefit from EGFR, can we actually take an FDA-approved drug, right, that we've all been disappointed in, and select the subgroup that we think that will be responsive? And so we're using an adaptive design where patients are going to be subtyped at UNC Health. This is now in the metastatic setting, but patients. So if anybody wants to do this in the neoadjuvant setting, we would be totally excited about it. But so patients will be subtyped, and if they're basal, that is the group that is going to be getting an EGFR inhibitor and getting erlotinib. So these are just 2 use cases of how we can bring integral markers to the clinic, and hopefully that inspires you to have other creative ideas to bring your biomarker or other biomarkers that you think might be predictive into the clinic. And thank you. That's it. Clearly, a lot of work takes a village. I'm presenting a lot of work from a lot of collaborators, but hopefully this is interesting to all of you.
Wow, Dr. Ye, that was a real tour de force. I mean, that was a great talk. I really appreciate it. And sitting in the background here, texting with my co-moderator, us how awesome this talk is. So really appreciate the critical information and the opportunity to learn. I have a question, if you don't mind, as someone who's also interested in integral biomarkers with ctDNA, is I find it to kind of blend the idea of integral markers and endpoints here, interesting to see that a lot of these, I think, lend well to early phase trials because there's no randomization, right? You're actually selecting based on a tried and true sort of finding that we obviously saw with the basal and classical like subtypes, right? So at the end of the day, if you find out within the context of a trial where you go in the fork in the road and get your therapy that we think you should get, that it actually looks like people do better that way, do you then believe that we have to examine that sort of thing in a randomized trial to prove external validity with an endpoint of truly survival? Number one. And number two, if you were to do that, do you think that it is ethical and there's truly equipoise to run that study if you already know that half the patients are going to do poorly when you randomize them?
Yeah, I think you bring up a fantastic point. I think that whether you randomize your biomarker, right, that looks promising, somewhat depends on limitations of sample size, right? So sample size becomes a really big issue when you're talking about randomizing biomarkers, or so sometimes it's not feasible. And so, you know, for the EGFR trial, we're comparing against historical controls, right? Because there's no way, there's just no way, even with 87 or with 54 patients, we need multiple centers to enroll that many patients. So ethically, right, can we do that? I think that's something that we struggle with a lot. Certainly with both trials that I showed you, I think some of the pushback that we get from reviewers is, even when we're selecting treatment, what are we doing for the other group, right? We're just trying to answer a question for one group. What are we doing with the other group? And so I think in an ideal world, if you talk to a statistician, absolutely you need to randomize your biomarker, right? In an ideal world, if you have 2 different treatments that you can give, that would be ideal.
Totally.
Right? So you're randomizing, you're essentially doing like 2 separate randomizations on 2 treatment arms, right?
Yeah.
That's really, I think, when you have, that's when you really do no harm, I think. But imagine now that's like 4 groups, right? That you're that you're running. So I think maybe it's not feasible. For some tumors, I think it might be, right? But it's something that's really important to think about. And, you know, maybe if we all get our institutions together and do this, that we actually could do something like that. I also think that getting ahead of the question, right, and doing integral markers, whether it be ctDNA, right, or something else, is really important. Because what happens in these large trials is that, let's just say the RAS inhibitor trials, yes, they're doing randomized phase 3 studies now. But by the time the correlatives come out, we're 2 years down the road, right? And then we're looking at, we're going to design another trial, right, for prediction, right? So can we get ahead? Can we actually ask the question for ctDNA, right? And then we would be at least a couple of years ahead of most randomized trials coming out.
I think that just definitely gets to the impetus here, and all y'all on the webinar now. In the future, if you take up clinical trials, writing dynamic trials that you can keep building on the next one rather than a decade-long type trial is I think sometimes a huge disservice. But it's the same thing with ctDNA, is so you have a single arm trial and show that one thing is good and one thing doesn't matter, and then if you randomize then to bring that to the clinic, I personally think that's unethical, because you're exposing people to something you know doesn't work. But interesting point. We have some questions in the chat here. Dr. Chawla there, if you want to ask them.
Yeah, so Dr. Martel, question, Dr. Ye, you touched on this, but at a high level, when would a biomarker graduate from being, graduate from being a subtype differentiator in a clinical trial to an endpoint in and of itself as a primary endpoint in a trial, for example, in a large phase 3 study?
Great point with ctDNA. Just want to add that. Yeah.
So, you know, if we use the ctDNA example, right, let's just take colon cancer and ctDNA. If we use ctDNA to differentiate adjuvant therapy after surgery, right? Should the high ctDNA high patients, are they high risk, and should they be the ones to get adjuvant therapy, right? I think that when the signal's there, like you were saying, Dr. Newhook, you can do a trial where it's an integral marker. I think what Dr. Martel, if I'm correct, is asking an integrated, when a marker goes from an integrated retrospective marker to a predictive biomarker. And I think that's something, if you have a rigorous assay with a valid hypothesis that could potentially benefit a patient, we should consider putting it into an integral biomarker trial, right? And the assays, of course, you know, you have to do a lot, right, to show that the assays are validatable, right? So once you have a integrated marker where you see, let's just you see like a response, right? So patients who have high ctDNA getting adjuvant therapy in colon cancer do a lot better than historical controls, right? So now you need the company, right, or you, to have a ctDNA assay that you know gives you the same result every single time, right? Then you can go into a clinical trial, whether it be a phase 1, 2, or 3.
Dr. Ye, you know, your journey, you know, I've been following, and I know we've spoken, and it's amazing that actually you and Dr. Sai connected at a surgical meeting and are doing amazing things as surgeon investigators. It really is inspiring to, you know, to all of us to hear your story. You know, some of the questions that have been asked focused on how to, you know, develop an integrated, you know, endpoint towards your clinical trial with integrated biomarkers. I wanted to touch on a couple of things with regards to basal-like and classical, if you don't mind, with regards to pancreas cancer. When we think about the results of the COMPASS study, and you think about what we're seeing in terms of the metastatic disease space, you mentioned that 20% of patients with basal-like disease. Do we have a signal with regards to the local regional space? Is that looking the same? Curious to know, based off some of the studies that obviously the group in Canada is doing, and as well with you and Dr. Sai.
A signal in the space, meaning like response?
More so, the percentage of patients that are actually basal-like with localized disease.
So if you look at all comers, like 800 patients that have been sequenced, in the metastatic setting, it is higher. It tends to be, depending on the study you look at, it could be anywhere from 22 to 26%. I think at its very highest. Dr. Knox says it's 30% in their cohort. So definitely a stage dependence for that. All of our old data and, all of the sequencing data that has been done is largely on resected samples. So primary, locally resectable. And retrospectively, that's been about 19 to 20%, pretty stable. We don't know why, but the MCW pancreas trial and the Honor Health trial, their percentages have been a lot lower. And they originally were enrolling only resectable patients, and they've since made an amendment to open to locally advanced. So don't know yet, but for sure there's definitely basal patients. The question is, there have been a lot of studies, and I think Renfield has reported this too in his last paper, where people are, where we're thinking, well, maybe there's population shifts after neoadjuvant chemotherapy, right? And it probably depends on the drug. So that probably wasn't part of your question, but so I think that it's actually really important to know what it is at baseline. And I would push the AHPBA and those of you who are interested in clinical trials, like if you have a non-invasive biomarker, awesome, right? Like you can't do better than that. And I'll also pose like the question and the challenge that if ctDNA is really hard for some tumors, what about biliary fluid, right? Or something, right? That we actually think about a lot as surgeons, right? But others perhaps don't. So is there something that we can actually leverage? I sort of lost my train of thought, but so non-invasive biomarkers are great. Oh, but pre-treatment biopsies are incredibly important. And then whether it be on treatment. So let's just say you have it in neoadjuvant trial, right? That like Dr. Drenkow has. So you get your pre-treatment biopsy, must. And then a portion of patients aren't going to get to resection, right? And so to do a really rigorous trial, you actually need everybody to have on-treatment biopsies, right? Not just patients who get resected, because that's just a subpopulation. And it may or may, it's going to skew, I think, confound your results. So I think your points are incredibly important, and that we as a field, I think, are uniquely positioned to think about that.
That's really interesting, all of those points, and particularly, that pre-treatment biopsy. And obviously the NeoPanc-1 study is going to highlight some of the feasibility of that. That being said, in your field at UNC, you guys have such a great infrastructure to have that pre-treatment biopsy. For clinical trialists that across the country, how have you leveraged your community, your structure at your institution to get those biopsies, those crucial biopsies for disease like pancreas cancer?
So I think that at the end of the day, it's all about collaborations, right? And team science and teamwork. I will say that I would propose that other institutions are probably better than us, and we actually learn from other institutions. But I think that, working as a team and having everybody sort of interested in a single mission is incredibly important, right? So I think one thing about, depending on where you are, having your gastroenterologist enterologists, right, do your EUS biopsies, they need to be like invested and interested and be willing to do those, to do the 22 cage chart cores that most institutions that are doing pre-treatment biopsy have shown are safe, right? And so providing like 3 extra cores, as long as they think it's safe, right, is okay, right? And so I think we as a field probably need to talk with our colleagues and the community and make sure that that is something that I think once you try, right, and educate, then it's okay. And I know that just from talking to colleagues who've run these trials, I've also heard that when you run the multi-centers, right, some centers are much harder. It's much harder for them to get to adopt something where the biopsies can be high quality biopsies that are actually usable. So that, for sure, that is absolutely an issue. Honor Health has been fantastic. So I think it's really getting, just having everybody work together and be willing to understand that it's like really, really important. And so how can we get there, right, and answer the question together?
That's great that you've been able to do that. Just piggybacking off of that, when you mentioned earlier the importance of the neoadjuvant space, and certainly as surgeons, this is an important area for us to investigate in. Most of the studies that we see, obviously, in the cancer space start in the advanced and metastatic space, and I am a true believer that the neoadjuvant space is really unique in that we're able to look at response and some of these correlatives. And also the drivers of the cancer are going to be much different in the early disease space. Can you expand on your insights there, and how we as surgeons can push for more novel studies in the perioperative space?
Yes, I think that paradigm does need to change, right? Because just because something is effective in the in the metastatic setting also does not mean that it's effective in the primary setting, right? So that it goes both ways, right? Scientifically, I would venture to say, though I don't know this for sure, but I'm thinking of a couple of tumors, pancreas being one of them, but I think cholangio is the same thing, right? We know for sure that the tumor microenvironment in the primary and the metastatic sites is totally different, right? Like for pancreas, we actually know, I think Anirban Mitra, I don't know if his paper is coming out yet, but there's certainly a lot of groups that are looking at liver metastasis versus lung metastasis in pancreas cancer. And you know what? If you have a lung metastasis, you're actually sort of like, we know clinically, right, that people act differently, but biologically, they're actually different, right? So if we want to give the same therapy for somebody with a lung metastasis and a liver metastasis, probably not the same, right? Those tumor microenvironments are also different. And like you said, the drivers, right, could be different. And Dr. Iacobuzio at Memorial has done really nice evolutions to show that. And so understanding where you start with is really important. And I think a lot of us look at retrospective studies where we're looking at resected treated samples versus like never treated samples. And that's hard, right? Because we're not necessarily comparing apples to apples. So I think that, like Dr. Trankow said, in talking to industry, I think we really need to show them the data. Like it does not make sense sometimes, right? To do that. And companies are really, I think, hesitant to go into the neoadjuvant space, right? So that's really hard. And that's where sort of IITs come in, and maybe using ECTN or CTEP, right, is really helpful. Or small companies, right, who need a way forward.
Well, that's amazing. Any other questions from the chat? We have a few more minutes. I know we really appreciate your expertise. Again, both speakers have just been phenomenal. I think one of the best sessions I can remember today. So very much appreciate both of your insights and expertise. And I think that, you guys are really paving the way in this field as surgeons, and I truly mean that. And congratulations to both of you.
Thank you for having us. It was awesome. So
Much.
more clinical trials from surgeons.
Definitely.
I think, Jenny, this is really fantastic. I wanted to just maybe finalize with like one last comment. And I think for those who are, you know, aspiring sort of up and coming, not just scientists, but also, trialists, how do you start, right? I think, I mean, it's one thing to look at your career, which is phenomenal. And, you've had so many successes, and now obviously, and 100% like deserved, right? You're getting access to all these different tissues and stuff. So I think for someone who's starting super low and like doesn't know, or they're just like, oh, I'm like excited about a clinical trial, like it's like, how do you even start? So what would be sort of your piece of advice to someone who is like interested, right, in correlative science, but may not have all these resources and even mentorship and whatnot? I think that would be really great too, if you could answer that question for anyone on the webinar or anyone who sees this later on video.
Sure. I'll just say that I was young once.
Yeah, no, I'm sure you. I mean, I
And
think it's
didn't
kind of
have
what are the
resources
pieces? What would you do
to start
differently?
with.
Or, I mean, you don't need to have done anything differently because it's worked out for you perfectly. But,
But I do think that, well, I think number one, I think surgeons are surgeons as clinical trials. I think that while I think that we should train more and have more, right? There's more than there used to be. So that's fantastic. But I also think that partnering and working as teams like across institutions is really important, right? Because many times, unless maybe you're at MD Anderson, but or Memorial, like we don't have like all the resources, right? To answer the questions that we would like to answer. But if you have an idea, and maybe the AHPBA Clinical Trials Committee is like what the committee should be doing, right? Is to bring all these heads and bring all these ideas together, right? And essentially, well, I guess it's sort of like doing ACOSOG again, but I think it's really, really important to get together as a community. And I will say personally, when I started, I had like, I had a really naive clinical question, and my samples, very few of them actually were from UNC. So some were from Northwestern, from Evanston, from University of Nebraska, from Hopkins. And so it was really working as a community. I think half of those samples came from surgeons who are really interested in clinical questions, and the other half came from either medical oncologists or researchers, right, who are really willing to sort of pool their resources together to say, hey, this is a question that we're interested in. And I think the other thing for somebody who's beginning is to share, right? Because collaborations go both ways. And as you want to create these teams and successful teams and get these ideas together for your clinical trial, right? You want to be sharing and giving back, right? And paying it forward. And so what is the next step? Like, how can you get to the next trial? But I do think that it's a position that the committee is in to do.
Yeah, that's actually a really awesome, not just answer, but also maybe gives us as the clinical trial committee, maybe some impetus for, creating like a work group, based on like maybe disease sites, and giving opportunities for those who are really interested. Because I do think, like Tim and Akil mentioned, like, we have like several of us have very similar sort of interests, like whether it's in ctDNA driven world, like Tim and I have been like trying to in the colorectal liver met world. And I do think that we all kind of, I mean, like struggling is maybe not the right word to say, but, in silos a little bit, right? Like every group comes with, their little findings, and like, wouldn't it be so much more powerful if we actually had larger numbers? And because I do think, honestly, progress made probably will be a lot faster, and then impact is probably much more, effective that way. So again, thank you so much. This was wonderful. Thank you both, Dr. Newhook and Chala. This was really a great session, and we look forward to the next one. And for those who are on the webinar, we will send out questions and quizzes, so look out for those, so that you can get your certificate. Thank you so much, everyone.
Thank you. Have a good night, everybody. Thank you for listening.